Okay. Alkystis, Rene, if you want to go.
Yeah, I think I'll start. Shall we go, Sylvia?
Yes, please.
Yeah, okay. Good afternoon, everybody, and thank you for choosing to attend this VHF workshop. I'm quite happy today to share with you our efforts to develop and apply imaging agents to study cardiovascular diseases. Now, the roadmap to develop imaging agents and validate them in vivo, it's, as you see in this diagram, a multi-step process and a multidisciplinary process that really starts from identifying a biological target, the presence of which signifies a particular pathology or hopefully the severity of that pathology. And it's really the identification of biological targets is really rooted in understanding the biology of the disease that we are studying. And then after we identify a target, we turn to our chemistry knowledge and to our chemists, to our colleagues, the chemists, who help us identify, synthesize, screen, and characterize compounds that have the potential of binding to the biological target that we're interested in. And after all of these experiments, we hopefully have good, promising compounds to bring forward for preclinical testing in different animal models. And then eventually, if the preclinical experiments are promising, we can then start considering the possibility of first-in-man clinical translational studies and full phase 1, 2, and 3 studies that may lead to launching a product into the market. Now, just to give you a feeling of how long these processes take, typically from identifying a biological target all the way to performing sufficient and convincing preclinical animal experiments, takes about 5 years and £1 to £2 million. Then to do phase 1, 2, and 3 clinical studies takes another 7 years and £10 million. And finally, launching a product into the market and getting regulatory approval takes another 2 years and £1 million. Now, the reason why we want to develop imaging contrast agents is to help us understand the biology of the disease as it happens in situ, in the tissues, and in vivo, to help us identify new imaging biomarkers to stratify the disease, tailor treatments, and understand responders. And also, we can use the platform of molecular imaging in preclinical models to test the efficacy of new and existing drugs. Now, if we go kind of step by step through this roadmap, as I mentioned before, initially we start by identifying a biological target. And in reality, there are abundant techniques out there, biological techniques, such as proteomics, genomics, metabolomics, microscopy, molecular biology, and high-throughput assays, all of which can help us to identify proteins, genes, metabolites, and pathways that are upregulated in certain pathologies. So, for example, for our own work over at St. Thomas's, we used simple histology to study the remodeling of extracellular matrix proteins in the context of cardiovascular diseases. And using tissues from different animal models and biopsy samples from patients, we observed that there's an active elastin and collagen remodeling in different cardiovascular diseases. So, for example, here you see tissues stained with elastin, that there is new elastin fibers within the growing plaque. There's elastin in the dilated aorta, and also there's elastin in the remodeled myocardium. Similarly, when we stain our tissues for collagen, we observe that there is an abundance of collagen 1 and collagen 3 within the plaque, within the dilated aorta, and within the remodeled myocardium. And fortunately, these fibrotic proteins are expressed in high abundance, which allows us to perform molecular detection with MRI. But also what we want is to kind of push the envelope in our understanding of, let's say, in our case, extracellular matrix turnover in cardiovascular diseases. So if we take a closer look into the biosynthesis of these proteins, we know that in controlled tissues, we have cross-linked elastin and cross-linked collagen. We also know that the synthesis and maturation of these fibrotic proteins occurs during the late fetal and early neonatal periods of life, after which the synthesis ceases. Unless there is some kind of vascular pathology or tissue injury that triggers two processes to take place. First, we have enzymatic degradation of these proteins, leading to accumulation of both elastin and collagen-derived peptides. But also we have active de novo synthesis of these fibrotic proteins in the form of tropoelastin monomers and collagen triple helices. Now, the fate of these proteins depends on the expression levels and activity of the enzyme lysyl oxidase that catalyzes the formation of these cross links, as you can see here. So you can imagine that if lysyl oxidase activity or expression is downregulated, that leads to the deposition of not cross-linked tropoelastin monomers and non-cross-linked collagen triple helices. Conversely, if there is excessive activity of the enzyme lysyl oxidase, this may lead to over cross-linking of our fibrotic proteins. Now, when we reviewed the literature specifically about the elastogenesis pathway in the onset of cardiovascular diseases, we found limited information primarily from these two studies that showed that the majority of newly developed or synthesized tropoelastin monomers fails to cross-link into the polymeric form of elastin in human atherosclerotic plaques and also human aortic aneurysms. And also from a study in hyperlipidemic pigs, they observed downregulation of the lysyl oxidase mRNA levels, which kind of set the hypothesis in our minds that there might be a shift towards accumulating tropoelastin monomers in disease tissues compared to having elastin cross-linked in controlled tissues. So to verify what was published in the literature, we perform our own biological experiments using rabbit aortas, where we also observed that controlled tissues have, as expected, multiple cross-linked elastin fibers, but they don't have any tropoelastin, whereas disease rabbit plaques have a highly abundant network of tropoelastin monomers. So in combining what we know or what we knew from the literature and with our own experiments, we have identified at that point tropoelastin as a potential new imaging biomarker, and we set out to develop a tropoelastin-specific imaging contrast agent as part of a BHF program grant. Which then brings us to the second step of this roadmap, which involves identifying, screening, and characterize potential compounds that have the ability to bind to tropoelastin. And of course, there are, again, a lot of techniques that one can use to perform this task. For example, phase display assays, through chemical modeling, through protein-protein interaction studies with crystallography and NMR, solid phase binding assays, and so on. But all of these techniques can help us to identify the compounds, synthesize them, and effectively label them for our imaging experiments, define and screen the specificity and their binding affinities, characterize the MRI relaxivities in case when we want to develop MRI contrast agents, and lastly, study the stability of these compounds in blood and plasma. So for our tropoelastin projects, let's say we chose a kind of a shortcut to identify potential compounds that may bind to tropoelastin. And with that, I mean that we dig into the literature and we looked for protein-protein interaction studies between tropoelastin and other proteins that interact with tropoelastin, such as the elastin-binding protein. And specifically, we wanted to check whether the interaction site and the specific amino acids involved in this protein-protein interaction were described, because our hypothesis was that we could take this amino acid sequence, have it chemically synthesized in the lab, and turn it into the target moiety of our contrast agent. So if we take a closer look into the way that we like to design our contrast agents, is for them to have a small molecular weight peptide up to, let's say, 16 amino acids that binds to the target protein, and onto which we attach a dot alkylator at the N-terminal lysine. And the reason why we need the dot alkylator is because we can use this cage to coordinate elements such as europium, gadolinium, and radioisotope such as gallium-68 for our in vitro assays and in vivo molecular imaging experiments. But the important thing here is that we want our contrast agents to be of low molecular weight, because in this manner they have favorable blood biodistribution pharmacokinetics that I will describe a bit later. So the screening and characterization of candidate peptides really aims to select from a pool of candidate peptides the best available ones to bring forward for in vivo molecular imaging experiments. And as I mentioned before, first we need to be able to synthesize our peptide with high purity. We need to be able to label the peptide with the appropriate element depending on the study and experiment we want to perform. We need to be able to do solid phase binding assays to screen for the selectivity of our peptides and determine their KDs, their binding affinities. We need to do HPLC experiments to test their stabilities in plasma and blood. And for gadolinium MRI experiments or MRI probes, we need to be able to determine the R1 relaxivity of the peptide, free in solution, and after it's bound to the protein of interest. And I will discuss this a bit later as well. Now on this slide, I will share what kind of results one can expect to get from an in vitro binding, solid phase binding assay. So for example, in our case, in order to develop the tropoelastin agent, we had two candidate peptides, peptide 1 that is abbreviated as the YPDH, and peptide 2 that is abbreviated as the VVGS, both of which were identified, as I described before, by protein-protein interaction studies published in the literature. So what you can see here in this in vitro binding experiment is that both peptides bind to tropoelastin. However, only the VVGS peptide showed the best discrimination between the binding to tropoelastin versus binding to mature polymeric cross-link elastin. The second thing we can observe is that both peptides bind to other extracellular matrix proteins, such as collagen 1 and fibronectin, but to a significantly lesser extent. We can also see that scrambled versions of each one of the peptides, or the DTPA chelator alone without the peptide, do not show any binding to any of the proteins, meaning that we need to have the peptide, and we need to have the peptide with the specific amino acid sequence in order to have some form of binding. And also what we get from the in vitro binding assays are the KDs for this interaction, which we hope to be as low as possible, because in that way our agents are more efficient in binding to the target. After that, we want to verify that the peptides we chose actually do co-localize with the protein of interest in tissues, either from animals or from humans. So we performed the co-localization experiments using rhodamin label peptides and immunohistochemistry. So for example, in this case, one of the two peptides, the VVGS, showed co-localization within regions of the aortic aneurysm that were deemed to be tropoelastin rich, as detected by immunohistochemistry. However, there was no co-localization between the peptide and tropoelastin negative areas, which were, however, rich in mature elastin, as you can see here. So these experiments kind of validated also the in vitro solid phase binding assays. And the third set of experiments we typically perform for gadolinium MRI contrast agents are to define the bound and free R1 relaxation rates of these peptides. So in this case, we measure the relaxivity of the peptide in an oscillating magnetic field when the peptide is free in solution and after we add our targeted protein. And what you can see is this characteristic increase in relaxivity in the presence of the target protein, which signifies a physical interaction between the peptide and the protein of interest. Now, after one collects information from in vitro binding assays, we may proceed to the third step of this process, that is considering possible animal models for in vivo experiments. So usually we like to start from small size animals, such as mice and rats, for the obvious reasons of being cost effective and easy to handle. And then if these experiments are promising, we may consider moving to medium-sized animals such as rabbits, and lastly to even big-sized animals such as pigs. And the real aims of preclinical imaging experiments are to, number one, define the biodistribution and pharmacokinetic profiles of these compounds. Number two, to study the toxicology and safety. Number three, to test whether the probes allow us to quantify disease progression and regression in vivo. And lastly, we can use these preclinical experiments as a platform to further improve the chemistry and optimize the chemistry of our newly developed probes. So in our case, and going back to the tropoelastin project, as I mentioned before, we had two candidate peptides, the YPDH and the VVGS, which we had, in this case, a radiolabel with copper-64, and we perform in vivo biodistribution experiments over time. So what we're looking for in an in vivo biodistribution experiment are, number one, the time that it takes for the probe to clear from the blood pool, because this defines the optimum time point of performing the in vivo experiments. Then we look what is the primary route of excretion of this contrast agent from the body, which could be either through the liver or the kidneys. So as I mentioned before, we design small molecular weight contrast agents because we want them to actively clear mainly through renal excretion. But occasionally, there is also some liver uptake. So for example, the YPDH peptide, although it was a small molecular weight compound, did show uptake in the liver, whereas the VVGS peptide did not. And that really depends on the lipophilicity of the particular amino acid sequence. The third thing we're looking in the biodistribution experiment is whether there is increased uptake or different uptake of the probes within tissues that carry the pathology that we want to study. And if you look at these bars here, you see that VVGS peptide did show higher uptake in atherosclerotic plaques in mice compared to the YPDH peptide. And lastly, what we want to see is no unspecific uptake in tissues that do not carry the pathology of the disease we want to study. So collectively, if one summarizes all the in vitro experiments and the in vivo biodistribution experiments, we concluded that between these two peptides, the VVGS one offers a superior, potentially superior contrast agents to study tropoelastin remodeling in vivo. And for this reason, we brought this agent forward for validation in different animal models of the disease. And in our case, we used 3 different animal models: the apoE-Muri model of plaque progression in the brachiocephalic artery, the rabbit model of plaque instability in the abdominal aorta, and the angiotensin II infused apoE-Muri model of aortic dilatations. So this experiment showed that using the tropoelastin agent in control animals, we didn't have any uptake. And that was because, as I mentioned before, control tissues only have cross-linked elastin, but no tropoelastin. So the lack of signal was actually good in this case. But as the disease progressed in the brachiocephalic artery of apoE mice, we can see an obvious increase in the area of enhancement and the intensity of enhancement, which signifies the accumulation of tropoelastin during atherosclerotic disease progression. And in fact, the MRI findings were validated by histology and also by Western blotting. We were also able to monitor treatment response using our tropoelastin agent, and here you can see that there is less enhancement in animals treated with statin. Then we wanted to advance our understanding of the biology of the disease and ask the question, does tropoelastin signal enhancement relates to plaque instability? And to do that, we needed to use a rabbit model that of plaque instability in the abdominal aorta. And also we needed to upscale the production of our contrast agent. So what we observed in this experiment was that indeed unstable plaques that progress to form, to rupture and form thrombi had a higher uptake of the contrast agent and a higher tropoelastin signal on MRI compared to clinically silent stable plaques. So this data probably suggests that tropoelastin does get increased during disease progression and can potentially be used as a marker of plaque instability. And then we wanted to further validate the use of this tropoelastin agent for other pathologies. So we wanted to ask the question whether dysfunctional elastogenesis also occurs in aortic aneurysms. And for this study, we used the angiotensin II infused ApoE mouse model, where we observed that the tropoelastin signal does correlate and does confine within the area of an aortic dilatation, but there is no tropoelastin enhancement in non-dilated aortas. So as I mentioned before, the preclinical experiments can also be used to help us further improve the chemistry of our contrast agent. And in this study, in collaboration with chemists at the University of Turin, they help us to turn our tropoelastin monomer into a tropoelastin tetramer. So as you can see from the schematic, the tetramer carries one tropoelastin peptide, but 4 gadolinium atoms at the end terminus compared to 1 gadolinium that we had in our monomer. So you can imagine that we can, in this scenario, we can deliver more gadolinium within a single binding event, which can potentially increase our signal, extend our imaging acquisition window, and potentially reduce the injected dose. Also, the specific chemistry that the team in Turin uses potentially has the ability to increase the thermodynamic and kinetic stability of the agent without causing any additional steric effects. So after they improved the chemistry and they synthesized enough of the tetramer for us to test, we went back to the animal experiments, to the murine model, and we performed a head-to-head comparison between different agents, an elastin-binding contrast agent, our tropoelastin monomer, and the tropoelastin tetramer. And what we observed was indeed that the tetramer tripled our contrast to noise ratio and doubled our imaging acquisition window. And also, using HPLC experiments, we found that the tetramer has increased stability in serum and blood, and it's stable beyond the 2-hour window. So collectively, we were able to produce a second and improved generation of the tropoelastin contrast agent with favorable imaging characteristics. Now, after a lot of in vitro experiments and preclinical validation in different animal models, one may consider to move forward for first-in-man clinical translation trials. And what we're looking at in this phase is to test whether the target is still present, whether we can safely upscale the production of the probe in sterile GMP conditions to be able to inject it into humans. We need to determine the pharmacokinetics and the dose for humans. But in our case, in order to also facilitate this clinical translational step and strengthen the clinical relevance of our work, we also collaborated with Professor Smith and Prakash Saha at KCL, who gave us access to carotid endarterectomy specimens. And what we did with these specimens was to image them at least ex vivo before and after soaking the tissues in the tropoelastin agent. And what we observed with MRI was that indeed the agent was bound to the tropoelastin in the carotid plaques, and we had an increase in R1 relaxation rate, signifying that potentially the peptide still has the ability to bind to the human version of tropoelastin, not just the murine and rabbit version. And also that tropoelastin itself is present within these carotid tissues. So going forward for a first-in-man clinical translation, we potentially have 2 avenues to explore. We can either use molecular MRI or nuclear molecular imaging. And of course, there are advantages and disadvantages in each one of these techniques. For example, if we go down the pathway of molecular MRI, we can take advantage of the higher resolution and soft tissue contrast that MRI offers. But we will need to use high dose of the contrast agent because MRI is not as sensitive, which could potentially make our project or the production of the probe more expensive and require further toxicological studies. On the other hand, if we choose the nuclear molecular imaging pathway, we don't get such a superior resolution as with MRI, but we can take advantage of the higher sensitivity of this technique, which then allows us to lower our injected dose, making the production of the agent more cost-effective and requires less extensive toxicological studies. So in reality, choosing the right modality will depends on each project and what we would like to study. So just to conclude my talk, I hope I gave you a flavor of the steps involved in developing and validating imaging probes. This process really starts from biomarker discovery. Then we proceed to compound identification and screening. We need to do our preclinical validation in as many animal models as possible to make sure that we are imaging what we think we're imaging. And then if this is successful, that could potentially lead to clinical validation studies and launching a project into the market. And so, as I mentioned in the beginning, this is a process that takes many years, and it's an endeavor that requires quite a lot of funding to complete. So I think with that, I would like to thank you for your attention and welcome your questions if you want to discuss them now or later.
Shall we move on to Rene's presentation and then take question all at the end?
Yes, for me.
Thank you. Rene?
Yeah, I'm here. Sorry,
Fantastic.
just had to get oriented. So let me just see. I'll get my presentation over here. Can you see my presentation?
Yes.
Okay. Perfect. So, so I'd like to talk about MR physics and applications, and I will mainly focus on cardiac MRI because this is what we are doing in our laboratory, and good. I don't know whether the screen is flickering. It's just flickering a bit on my side, but hopefully it's clear for you.
Looks fine.
Perfect. So as Alkystis already said, let's say the whole experiment usually requires to think about target screening, the chemistry of developing a contrast agent, of course, then having the right animal models to validate the new tracers. But then also we need to have the right imaging technology, data processing, as you mentioned before. And ideally, at the end, we would like to somehow impact the patients. And in this presentation, I only want to talk now about the imaging technology, and I want to focus on MRI. So as I said, we can use multiple imaging technologies, either to scan patients or to scan animals. So basically we have the anatomical methods like CT and X-ray, but also ultrasound. And then we have the molecular imaging methods, PET and SPECT, and but also some more advanced methods like magnetic resonance spectroscopy or hyperpolarized MRI. And on the preclinical side, we also can use optical imaging which has a small penetration depth. That's why we don't use it clinically, but in preclinical models that can be quite useful. And also what people nowadays are getting more and more into is photoacoustic imaging. So looking at the electromagnetic spectrum, we can differentiate the different imaging modalities in ionizing radiation and non-ionizing radiation. And as you can see, most of the techniques, X-ray, CT, and PET, are basically using ionizing radiation and can be harmful to the patient. However, MR basically uses non-ionizing radiation and is basically on the left-hand side of the spectrum. So the most that can happen is that there can be some tissue heating if you are not careful enough. So that differentiates these technologies. So MR is fairly non-invasive, whereas, let's say, I call these other techniques invasive, even so we may not even introduce a catheter, but mainly because of the ionizing radiation they require. So if we compare now the different imaging modalities, and as an example, I just want to look now at coronary angiography. X-ray remains the gold standard, provides high resolution images of the coronary arteries, and one can do diagnosis and treatment in one session. But as I mentioned, it's invasive, potentially risky for the patient, and there's radiation exposure and also use of contrast agents that can be nephrotoxic. And of course, it's expensive and provides limited information on the extent of arteriosclerosis. And that is the main interest in our lab. And that is the reason why we are not using it. So basically it provides little information on plaque burden, plaque composition, or vulnerability. And then there's CT coronary angiography, which is very fast. It can be performed in a single breath hold, and it's easy to perform, provides high spatial resolution, about 0.5 millimeter isotropic, and has a very high negative predictive value. So basically can be perfectly used to rule out coronary artery disease. And currently it's recommended for patients with low to intermediate risk of coronary artery disease. But as I said, there's radiation exposure, need for nephrotoxic contrast agents. There can be also blooming artifacts from calcified plaques, so they can, for instance, hide the stenosis, so that is a problem. So even so calcification on CT is highly diagnostic, it can prevent from proper diagnosis of a coronary stenosis. And there is limited information, for instance, on myocardial perfusion and viability or LV function, which is a strong area of MRI, and I will talk more about this. But of course, there's another technology available, especially for molecular imaging, and that's the clinical gold standard for molecular imaging. That's Positron emission tomography. Has a very high sensitivity for radio tracers, provides quantitative data, and allows molecular imaging, for instance, of proteins. We can do cell tracking. And this is an example here of a sodium fluoride scan where we can see microcalcification in the coronary arteries, which also has been shown to be predictive of future coronary events. But the disadvantage of PET is the resolution is fairly low. Again, there's ionizing radiation, and of course one needs a radiochemistry lab and ideally also a synchrotron. So it's a very expensive technology, and compared to SPECT, PET is not multiplexing. So coming now to MRI, the technology that we are mainly working in our laboratory. So it's non-invasive, no radiation exposure. There are no known side effects unless one uses the contrast agents. And it enables lumen and plaque visualization. On top of it, also it provides anatomic and functional information of the myocardium and allows for tissue characterization. For instance, we can identify a fibrosis and edema, but also iron deposition or fat fraction and whatever we encode in the MR signal. And this is just an example here. We see a coronary artery with some stenosis. So it's also basically becoming more mature for coronary artery imaging, which is our long-term goal. let's say in our lab, apart from, of course, my cardio tissue characterization. But also there are disadvantages. It's sensitive to cardiac and respiratory motion, so the scans are usually taking much longer, so we have to take care of that. As I said, we have long acquisition times, and we have limited resolution. I mean, compared to PET, the resolution is very good, but compared to X-ray and CT, the spatial resolution is limited, about 1 millimeter isotropic. Also, currently we are pushing it to 0.8 or 0.7 millimeter isotropic in the heart. And so talking about MR, I think we also need to know basically what are the components that we need to make, basically to create an MR image. So first of all, we need a very strong B0 field. So basically when we talk about 1.5 or 3 Tesla or 7 Tesla, this is the B0 field. And that is being used to polarize the spins, basically to polarize the spins of the protons. But then we also have to basically excite, or basically we have to flip those spins once they are polarized. We do this with a B1 field, or we call it a rate of frequency field, or we can call it a spin exciter. And finally, we need a third magnetic field. It's the gradient field that we use for spatial encoding. And the B1 field also can be used to receive the MR signal. So basically we can build basically send receive coils, or we can send with the body coil. But anyway, so we need coils to receive our MR signal. And then of course we need computers to basically process the data. And what I show here is a pulse sequence. Basically, this is the temporal flow diagram, how we play out these radio frequency pulses. This is this RF, and how we play out the gradients for spatial encoding, which is basically here number 4. These are the gradients, and we use this for spatial encoding. And then what we do is we don't acquire the image directly, but we acquire K-space, so it's a frequency space, and then we perform a Fourier transform to get the image. So basically this process, this is the space that we are measuring. We measure line by line, and by performing a Fourier transform, we can get a high resolution MR image. So the signal we are getting in MR usually comes from fluids. So basically what we image is water. So we get signal from blood, body fluids, or CSF. But we also get signal from muscle, fat, and white and gray matter. But of course, they contain water, or let's say they contain hydrogen, and for that reason we get a signal. And no signal or low signal we get from hard tissues such as bones or ligament or teeth, or for instance from gases from the lung airspace or from the sinuses. So these are areas that appear black on an MR image. And here you just see basically some examples. So basically we can acquire basically cardiac function, or do a perfusion scan, or also can do flow. So in the next slides, I want to show basically how we basically get the MR signal. So basically we have to use this strong magnetic field, B0, that aligns the spin of the protons that act like little magnets. And if we align them all in the same direction, we can create a net magnetization. And this net magnetization basically determines the signal to noise. Now, once we have this magnetization, and if this is in a magnetic field, it starts precessing. So we call this the Larmor precession. And there will be more about it. So that's very important in MR. So this is basically, in a way, the equation of motion. And with some extensions, we call this the Bloch equation that tells us how our magnetization vectors move. Now, in order to measure a signal, we have to flip this magnetization vector that is now aligned with the main magnetic field into the transverse plane. And now, because of the B0 field, it starts precessing. And we can see this here. So basically now in the presence of the B0 field, this magnetization precesses and induces a current in this coil that we can measure. And it decays with a certain constant, which we call T2. And that's also a contrast that we can generate with MRI. Now, under these conditions, let's say if this was the image that we're trying to image, and let's say each circle basically is a representation how many protons we have in this voxel, then if you only have B0, all the spins will precess with the same speed, and that means we can't really spatially encode, let's say, the signal that we are receiving. So what we have to do is to, as I mentioned before, add a third magnetic field, which we call the gradient field. And we see this over here, this GX. And now in the presence of this gradient field, the spins to the right-hand side now precess faster than the ones on the left-hand side. And this way they have a higher frequency, and this is the way how we can differentiate now between spins that precess at lower and higher frequencies, and that we use for X encoding. And we can do the same in the Y direction and the Z direction. So basically by basically having different precession frequencies, we can spatially encode our signal. And this is just in a nutshell how we do it. And you can see here basically now the equation is B0 plus this gradient. So basically we can turn on and off these fields and basically change the way our spins are going to move. And as I mentioned, our signal decays with the T2 relaxation time. This is something we can see here on the lower part. I don't even know whether you can see I think maybe now you see the laser pointer. So basically our signal decays with T2, that's the T2 relaxation time, and returns basically to the main magnetization along the main magnetic field with T1. So these are the two relaxation times that we can take advantage of, and that allow us to generate contrast in our images. So now talking about cardiac MR, so what I showed before is basically we can image the coronary arteries, we can even see a plaque, we can basically get cardiac function, myocardial perfusion, we can do viability imaging, and we also can do some tissue characterization. And I will show in the next few slides in a bit more detail how we can do this. For instance, this T1 mapping, we can map diffuse fibrosis, or with T2 mapping, we are more sensitive to water, so basically we can measure edema. And what I showed before, what I tried to explain, so the acquisition sequence is basically the explanation I gave before. So we have a certain pulse sequence that consists of this B1 field and the gradient fields that allows us to acquire images, but then we also can employ some pre-pulses that we can use to add additional contrast to our images. And we also have the option, for instance, to measure motion. We do this as navigators, but I won't go into this in this presentation. And of course, there are different flavors of imaging sequences. Again, I don't want to go into all these flavors, but there's a huge variety how we can acquire the data in MRI. We can also acquire them on a Cartesian grid, on the radial trajectories, biotrajectories, whatever we choose. So there's a lot of degrees of freedom. And I want to talk about now the pre-pulses that we have, the T2 prep and the inversion, because they're the most important ones, and they allow us to get either T1 and T2 contrast. So let's start with T2 preparation. So here, basically, we have now blood, muscle, and fat, and they have different T1 relaxation times and different T2 relaxation times. And now if we produce a T2 weighting in our images, you see we can differentiate between blood and the myocardium. So basically, the signal of myocardium decays much slower than that of the muscle, and this is shown here in this diagram. So basically, if we now perform this T2 preparation pulse, we can see that the magnetization of the myocardium decays much faster, and then basically the signal of the blood was decaying slower, and for that reason we can now generate this contrast between these two tissues, and this is shown in this diagram. So this is a scan with a T2 prep off, and this is one where we turn on this pulse, and you can see we have a stronger contrast between blood and myocardium. Unfortunately, here we are using a sequence that already has some T2 weighting. Otherwise, the contrast would be even much more flat, and you wouldn't see much difference between the signal of the blood and the myocardium. But nevertheless, I think it shows we can enhance the signal between the myocardium and the blood pool, and it allows us to create these nice images of the coronary arteries without the use of a contrast agent. And here in the lower row, you see, for instance, the same patient scan by CT, but of course with CT we have to inject a contrast agent. So I think that's a strength of MR, that we can do a lot of things even without the use of contrast agents. So another important contrast, as I said, is the T1 contrast. And again, here we can focus on, for instance, now myocardial infarction. Basically, the presence of a contrast agent has a very low T1. And here the normal myocardium has a T1 of 850. So basically now we can exploit the difference in T1 between normal myocardium and the infarcted myocardium. And this is usually done with this inversion pulse. Of course, we have to, in this case, we have to inject the contrast agent. And now in this example, you see the normal myocardium appears dark and the infarct appears bright. So why is this? So after we perform this inversion pulse, we invert the MZ magnetization, and now the tissue that contains the contrast agent that has short T1 recovers much faster, whereas the normal myocardium, which has little T1, basically returns much slower, and for that reason we get this contrast now between normal myocardium and the infarcted myocardium if we wait a certain time, which we call the inversion time. And this is just an example. Here you see now such an image, so basically normal myocardium, and here basically the infarcted myocardium with the contrast agent. And this is usually a late scan that we perform about, I would say, 10 to 20 minutes after contrast injection. But we also can do an early scan. So basically that would be this early enhancement. And there we also see the area at risk. So it depends how much time we wait. So if we wait 10 to 20 minutes, or we only see the necrotic core, but if we acquire the images early, we also see here this rim, which is still a salvageable tissue. So basically it represents the area of at risk. However, in many cases, the fibrotic disease in the myocardium is very diffuse, for instance, in amyloidosis or in, let's say, Fabry's disease. And here, for instance, on these LGE images, you see only basically normally looking myocardium. But then if you look at the T1 maps, in this case here, the native T1 map, you see basically much lower T1 values for Fabry's disease. So we see this here. So basically it has low T1 values. Or for instance, here for amyloidosis, we see fairly high native T1 values. But on top of it, we also can quantify the extracellular volume, which we call the ECV. And again, here in amyloidosis, we see a very high extracellular volume here in red. So we can see this here. And in Fabry's disease, we see a much lower extracellular volume here in blue. And the way we can quantify this, so I just want to demonstrate, so basically this is the extracellular volume. So basically in hypertrophy, we basically can have this increased extracellular volume between the myocytes. And of course, in myocardial infarction, that area is much larger. And that's basically the infarct area. So now the basic idea how we can quantify this is by measuring either the recovery of the MZ magnetization, and that allows us to quantify the T1 relaxation time. So basically we can do a pixel-wise fitting, and we know the model function, and that allows us then to get this T1 map. And for instance, this is the area of myocardial infarction, which basically has now a shorter T1, and this is the normal myocardium. And we can do the same for T2. Basically, there we basically now look at the signal decay of our MR signal. Again, we know the signal evolution. We can now do a pixel-wise fit to the measured data, so basically to those data, and create this T2 map. And in this case, for instance, here in this area, we have higher T2 values. Basically, we have a little bit of edema. So the T2 map tells us more about the water content. And this is just a summary of different diseases. And I also want to mention iron overload, for instance, which basically leads to very short T2 star values. So basically we can see this here, and which also leads to short T1 values. So basically that's one way how we can quantify it. And I mentioned already basically Fabry's disease and amyloidosis. And here, for instance, just now an example of a normal looking myocardium, the T1 map, the ECV map, and the T2 map. So I think this T1 and T2 mapping, including ECV, is a really nice tool to quantify diffuse fibrosis and edema in the myocardium. And the last technique I like to show is perfusion imaging. So here we also take advantage of the T1 relaxation time, but we don't use an inversion pulse. We just use a saturation pulse. So basically, we have the MZ relaxation, perform the saturation pulse. So basically, now the MZ is 0 for all the tissues. And now tissue with normal perfusion, basically where the contrast agent can get to, will recover faster, whereas tissue with reduced perfusion and basically less contrast agent uptake basically will have a slower T1 recovery. And for that reason, we can now differentiate between normally perfused myocardium and ischemic myocardium. So we generate this kind of contrast. And this is usually how it looks when we acquire those images. We acquire them over 30 to 60 seconds. And basically we can now basically measure the time course in the myocardium, basically of normal perfusion or, for instance, of an ischemic area. And we can do this, let's say, with eyeballing. This is usually what we do clinically. And also what I wanted to say is usually we only acquire 3 slices per heartbeat because we only can basically fit 3 slices into 1 heartbeat. So that's maybe one limitation of perfusion imaging at the moment, but there are new methods that allow us also to get to 6 to 8 slices and to almost cover the entire ventricle. And it brings me to my last slide. So as I said, we perform visual analysis, which is easy to perform in the clinic. So there's no complex analysis required. However, it requires expert training. So people have to train for a couple of months until they feel confident. So here an example where you see a diffusion defect, and that also can be seen nicely here on this quantitative perfusion map. But for instance, now if we have maybe microvascular disease and the perfusion deficit is more diffuse, then usually those maps are advantageous because they may be hard to quantify visually. Yeah, that brings me to the end. So I'd like to thank you for your attention, and I'm happy to take any questions.
If you have any question for Rene, please raise your hand or type it in the chat if your mic doesn't work.
Or for Alkystis.
Oh, that was me. Or for Alkystis, actually, both of you, sorry. It's the first time we have 2 presenters. The room is very quiet today. What do you think is going to be the next development in terms of imaging?
I think it depends now. I think that there's the, let's say, the physics side, the engineering side, and then there is the contrast agent side. So maybe I answer the physics side, and then Alkystis can talk about the contrast agents.
Sounds fantastic.
But on the physics side, I would say what we want to achieve, we want to make cardiac MR much faster, because at the moment we are competing with echocardiography, which is a lot cheaper. And especially when you look at the American market, the reimbursement is $500 for an echo and $500 for an MR. And an echo machine costs maybe, let's say, probably an expensive one, $250,000, and then MR scanner costs $1 million. So the business model is very different. So the only way we can improve it is to make the test faster. So the aim is to bring down the exam maybe to 10 minutes and to provide multiparametric characterization of the myocardium. So what we want is in one exam and even in one scan, get basically the information on fibrosis, inflammation, the anatomy, function. So ideally, we want to pack all of this in one scan, and that requires a lot of technological developments, such as advanced motion correction, image reconstruction. So basically, we have to acquire less data, but still reconstruct good-looking images. And what we do is we use techniques that you use when you compress an image, when you save a TIFF or GIF or a JPEG. You also reduce the size of the image from maybe a few megabytes to a few kilobytes. And that's what you do in MR, but we go the other direction. So we only want to acquire a few kilobytes, but get a megabyte image out of it. So these are some of the research directions that say the whole field is working on the engineering side.
Fantastic. Alkystis?
I think when it comes to imaging probes, our aim is to develop the most specific probes possible, the ones that hopefully bind only to the protein of interest and to nothing else. We want them to bind with high affinity and stay there for long enough for us to be able to image this pathology without rushing. To have a strong MRI signal if we were looking for an MRI probe. And then in the case where we have, let's say, a portfolio of agents, let's say for elastin, tropoelastin, or collagens, then we need to run preclinical experiments and test which ones and pick the best one, or pick the one that has the best predictive value or prognostic value for the disease. So one advancement is the chemistry side of things, and the other advancement is the biology of the disease. So which one performs better than the other? So we can improve on these two ends.
Fantastic. Is there any question coming from the audience? No, if there is no more question, I'll say we can call it a day. And thank you very much for your presentation.
Thank you.